The lichens are foliose, fructose, and crustose. The stems of algae, a stem is a stem, so I think stem site. The anchor would be hold fast, because you're holding the thing, or you're fastening the algae's tesserae, by the way. Okay, if a yeast divides unevenly, it's termed actually budding. If it divides evenly, it's termed fission. When you have a symbiotic relationship with one organism is going to benefit and one is going to be unaffected, that is. One benefits and the other also benefits, that's. One benefits and the other is harmed, is.
Parasitism.
There we go. Now, opportunistic would be, that can fall into the category of commensalism and mutualism. No, not mutualism. Parasitism, actually, when you get into opportunistic, it's like if you get that when you're immunocompromised. But opportunistic wouldn't be a symbiotic relationship. Okay, so zygomycota, sexual spores, asexual spores. So sexual, zygo. Okay, let's look at ascomycota. Sexual ascomycota would be ascospores, asco. So the phylum and the sexual structures are similar in name. And then the asexual would be Canidia. Basidiomycota have sexual spores called? Basidiospores. I didn't put this on a test, but what would their asexual spores be called? Canidia as well. Now, what are the symbiotic fungi with plants? Mycorrhizae. When you lack septa in the fungal hyphae, it is? Coincidic. Fungi, all of them. Resistant to mild pressure, requires low moisture, does not require a lot of nitrogen, and it can metabolize complex carbs. So fungi are decomposers. Obviously, they can break things down. Lignin is in bark, so fungi can break down trees. What did we talk about that didn't have mitochondria? Archaeozoa and Microspora. Microspora lack the microtubules, I believe, and they're in your eye. You can get the parasite in your eye. And then Archaeozoa don't have true mitochondria, they have something called a mitosome. Now, the flukes are typically monoecious. They would have a flat leaf-like shape of ventral and oral sucker, and on their skin is something called a cuticle. So it gives your worms a rigidity, but it's soft enough for food to come through. So it's all the above. Water moles have two flagella. They do look like cottony masses when they digest something, decompose something. They can reproduce asexually and also sexually, and are found as spores in a spore sac, but they don't have a single frontal flagellum eye spot. What does? You remember the euglenoids that we looked at? And they were green and they had this big red eye spot in them and one flagella coming out? Exotoxins are found where? Gram-positive. So exotoxins are made in the cell and can be secreted by the bacteria, but the bacteria doesn't die. Now, an endotoxin would be outside the cell, but embedded in the cell wall component, so you would have to destroy the bacteria to get that out. Fungal, okay, keyword here, deep infections, that would be, got it, systemic. The neurotoxin of a dinoflagellate is called a saxitoxin. Which of these do we get from brown algae? Algae. Cellulose and chlorophyll come from green algae. Carrageenan and algae come from red algae, and algae is the brown one. Protozoans reproduce via ovum, asexually, fission, budding, phylogeny, all of them. Short naked pieces of viral RNA are called viroids. They're called viroids. That will be a plant virus. Next one, this one. Western blotting, polymerase chain reaction, and restriction fragment polymorphisms are all used to detect viruses. It's just that Western blotting is more of a serological technique, whereas the other two are nucleic acid techniques. All of them. Okay, this one everyone had a question with. What is the chemical that your body is going to secrete to go to the hypothalamus? That's the answer. People would tell me super antigen. You don't make the antigen, you make the antibody. All of those enzymes can be used for pathogenicity. And how would you destroy an IgA? IgA protease. What protein is going to manipulate the cytoskeleton of the host? Invasins. Alright, this organism causes elephantitis. It's a helminth, is a worm. You can get a nosocomial infection via all of these things. Immunocompromised patients, transmission between staff and patients, just having microorganisms in the hospital. Okay. When a virus has to attach to a bacteria, the bacteria has to have receptors, and the virus has to have what molecule? Starts with an L. What's the molecule on the pathogen called that needs to bind to your receptor? In general, we call it a ligand, right? So ligands would be the molecules on the pathogen. You, as a host, you have a receptor. The ligand will sit in the receptor. But the thing is, bacteria, if a bacteria is a host, they have receptors all over them. Whether it's on the cell wall, or the fimbriae, or the flagella, you can have receptors all over you.
So it'd be all of you?
All of them, yes. All of these are found in viral nucleic acids. What's the mandatory component of a virus? A capsid or a protein coat, same thing. Okay, if you're going to grow a virus, you can use living animals, eggs, or cell lines. What's the period that you don't detect your viral infection? Eclipse or biosynthesis? Okay, when you have a clearing because of viruses that you grew in the lab, it's called a plaque. So when you have, okay, let's say you put a bacteria, a bad strain of strep onto blood, and there's a clearing, that's beta hemolysis. If it's green or partial, that's gamma. If there's no clearing, if it's partial, it's alpha. If there's no clearing, it's gamma. If you are looking at the clearings when you're dealing with viruses, it's called plaque. Multiplication of the viruses must begin with attachment. Well, if the virus doesn't attach to the host, how are you going to multiply the virus? Okay, the manner in which a disease develops is pathogenesis, because genesis means develop. seeing how it's transmitted is epidemiology. Getting the disease in a short space of time is epidemic. Endemic is a constant infection. Epidemic is a short-term infection that got spread in a short space of time. And if it's worldwide, it is okay, so we're looking at 50% of your test population. You want to see how many people are infected. It is ID 50. Viral infections in the blood are viremia. If I said just infections in the blood, it's all of them. But viremia would be viruses, toxemias, toxins. Bacteremia would be a bacteria, and septicemia would be a poison. Hypoparenteral. So the parenteral route, skin route, and mucous membranes, you can get, yes.